All right, today we're diving into a world that honestly sounds like it's straight out of science fiction. It's called synthetic biology. And here, scientists aren't just reading the code of life anymore, they're actually starting to write it. We're going to explore how they're engineering microbes to become these tiny, super powerful factories that could literally build our future. So let's just start with this one massive question. Can we program a living cell the same way we program a computer? I know, it sounds pretty wild, right? But this isn't just a hypothetical. It's a question that scientists are actively trying to answer right now.
So what's the biggest hurdle? Well, it's that natural cells are, well, they're kind of messy. You have to remember, they've evolved over billions of years with just one single goal in mind: survival. And that means they're jam-packed with all this extra genetic stuff, all these functions we don't need, which makes them really unpredictable and not very efficient for the specialized jobs we want to give them.
So how do you get around that? Well, scientists are creating something they call a microbial chassis. And honestly, the best way to wrap your head around this is to think about the frame of a car. It's the absolute bare bones foundation, something you can then build upon, adding only the specific parts you need for whatever job you have in mind. Okay, so this idea of a chassis really gets us to the core mission here. It's all about figuring out how to actually program life's code so we can build these tiny, specialized machines. Let's take a look at how they're doing it. The name of the game is simplification. That's really it. By stripping a cell down to only its most essential genes, you get a biological factory that's way more stable, way more efficient, and just predictable. All the cell's energy starts going into making the one thing you actually want it to make. But, how do you actually simplify a living cell? Well, it turns out researchers are taking two very different, almost opposite approaches to solve this puzzle. Let's break them down. So, on one side, you've got the top-down approach. This is all about taking an organism that already exists, like E. coli, and just strategically deleting all the genes that aren't absolutely essential for life. Then, on the other side, you have the bottom-up approach, which is, frankly, even more ambitious. This is about building a brand new, minimal organism completely from scratch, using only the necessary genetic parts. Let's go back to our car analogy for the top-down way of thinking. Imagine you take a regular old street car and you want to turn it into a race car. What do? You pull out the back seats, you yank out the AC, the radio, literally everything that doesn't make it go faster. What you're left with is a lean, mean, high-performance machine. So how does this work in a lab? Well, first, they use computers to analyze the entire genome and pinpoint all the genes that aren't essential. Things like leftover viral DNA or backup systems the cell doesn't really need anymore. Then, using amazing gene editing tools like CRISPR, scientists can go in and just precisely snip out all that genetic clutter. And finally, they test the new, streamlined cell to see just how much better it actually performs. Now, let's talk about the bottom-up approach. So if top-down is stripping down a finished car, bottom-up is like building a high-performance kit car from scratch. You don't start with a clunky, finished product. You start with a blueprint, and you assemble it piece by perfect piece, using only the parts you absolutely need for the job. And this process, it's just mind-blowing. It actually starts on a computer, where scientists design the perfect minimal genome from the ground up. Then, in the lab, they literally print that genetic code as a long strand of synthetic DNA. And finally, and this is the craziest part, they assemble that new genome and boot it up by transplanting it into a host cell, basically replacing its natural operating system with a completely man-made one. Okay, so that's all the theory. It sounds amazing, but is any of this actually working in the real world? Well, let's take a look at some of the incredible results that scientists are already getting. And just look at these numbers. Seriously, using that top-down approach, a stripped-down E. coli churned out 2.4 times more of a key amino acid. Another microbe boosted its enzyme output by 2.5 times. But here's the real showstopper. An engineered yeast cell increased its production of human growth hormone by a staggering 30 times. I mean, these aren't small improvements. These are absolutely massive leaps in efficiency. The bottom-up approach has led to achievements that scientists have described as nothing less than the dawn of synthetic genomics. And in what was just a landmark achievement, scientists built the very first self-replicating organism whose parent was a computer. They designed a minimal genome, they synthesized it, and they booted it up to create an entirely new species. I mean, just think about that for a second. It's a living cell that is running on completely man-made software. And it absolutely does not stop there. Take yeast, for example, a much more complex organism. It normally has 16 separate chromosomes. But recently, in this just mind-bending feat of genetic engineering, a team of researchers decided to see if they could, well, stitch them all together.
And what they created was a perfectly healthy, living yeast cell with just one single giant chromosome. This discovery just completely rewrites the rulebook on how life can organize its genetic information, and it opens up some wild new possibilities for engineering even more complex organisms down the line. So, these breakthroughs are obviously incredible, but you might be wondering, why does this matter to me? What can we actually do with these super-powered designer microbes? Well, the potential applications are just huge. I mean, imagine tiny biological factories that produce life-saving medicines that are just too complex to make with normal chemistry, or microbes that literally eat our waste and turn it into clean biofuels. We could design them to manufacture futuristic new materials, to clean up toxic spills in the environment, or even to help us grow more food. So where does all of this leave us? It feels pretty clear that we're standing right on the edge of a brand new biological revolution. And here's what's truly revolutionary about all this. Creating a minimal cell isn't just a cool lab experiment. Every single one of these breakthroughs teaches us something new about the fundamental rules of life itself. We're not just building better tools, we are gaining a much, much deeper understanding of what it even means to be alive. And that leaves us with one last really powerful question. We are learning to write the code of life. So the question is no longer, can we do it? But what should we build? What problems should we program these cells to solve first? That's something for all of us to think about.